Okay, so one of the big ideas of this chapter that is really important and really groundbreaking and kind of mind-blowing is, how did eukaryotic cells come about? How did eukaryotic cells get their chloroplast, get their nucleus, get their mitochondria, right? And that's kind of the big question that we're going to tackle with. But let's start out first with just, what is a eukaryote, right? We'll talk about the endosymbiont theory, but let's just talk about, what is a eukaryote? What makes a eukaryote a eukaryote, yeah? So eukaryotes are organisms that have a nucleus, right? That membrane that surrounds their DNA, and that nucleus, that membrane, that has pores. That way information in RNA can go in and out, and other molecules that are helpful for maintaining DNA, copying DNA, can enter and exit the nucleus as well, right? But it's still a protective layer around that DNA. Most eukaryotes have a mitochondria, or remnants of a mitochondria, right? Mitochondria being the powerhouse of the cell, right? So it's the organelle that makes ATP, right? But it's a membranous organelle. Eukaryotes have a very specific cytoskeleton, which allows them to shape change and be a little bit more flexible with the shape that their cells make, right? So when we talked about prokaryotes, they really only come in one of three shapes, right? They're kind of static, and really once they're in that shape, they're stuck in that shape. Eukaryotes are these shape changers because they have a cytoskeleton that is unique, that allows them to change the shape of their cells and have more variety in the shape of their cells. Most eukaryotes have a flagella or cilia. Some have lost it secondarily, and even within a multicellular eukaryote, not all of the cells will necessarily have cilia or flagella. Eukaryotes have linear DNA. So we talked about the DNA of a prokaryote being in a circle, right? Being one circular chromosome. In eukaryotes, now our DNA is in a linear strands that we call chromosomes, right? So different shape to our DNA, and eukaryotes tend to have a lot more DNA as well. Eukaryotes reproduce by mitosis as opposed to binary fission. Okay, so mitosis is slightly different because we got to divide the DNA in the nucleus, and then the rest of the cell divides. So it's a slightly different process, but it's the same basic idea of cell division. And eukaryotes are sexually reproducing organisms. They might not reproduce sexually all the time, but for most eukaryotes, they do have that option of mixing genes with another individual to increase the genetic diversity of their offspring. So what's so important about organelles, right? Prokaryotes, they don't really have an organelles. They have ribosomes to build proteins, and that's about it, right? In eukaryotes, organelles are super important because they help to increase the internal surface area of the cell, which allows the cell to do a lot of stuff, right? So most cells, especially prokaryotic cells, if they wanted more surface area, they'd have to make their outer cell membrane larger and larger and larger. That's a problem. If your outer cell membrane is larger and larger and larger, your cell just gets larger, you're going to increase the volume of your internal cytoplasm way faster than you'll increase the surface area. So not super good if everything you need to transport in and out of that cell is coming across that cell membrane. You want a large surface area to the external environment compared to your internal volume. Organelles are pretty nifty because they allow for that increased surface area but internalized, so that way we don't really increase the volume too much. We don't change that ratio of surface area to volume too much, but now we have more surface area inside to carry out stuff like building proteins, making ATP, photosynthesizing, right? We now have space inside to do that. It's kind of like having a closet with lots of shelves, I guess. I don't know. Versus just having a closet where you just like shove everything in and it's not organized. Closet with shelves, you have a lot more surface area. You can actually put a lot more stuff in there and make it a lot more functional. I hope that analogy made sense. Okay, so we've kind of seen this tree before. We've definitely seen this tree before. The three main domains of life: bacteria, archaea, and eukarya, right? Where do we put, if we were to put the trait of having a nucleus on this phylogenetic tree, where would we put it, right? Do we think it was here and then was lost? Where do we think it is? Yep, so only eukaryotes have a nucleus. So how did the nucleus originate? That's one of the key things about eukaryotes, is that they have a nucleus. Archaea bacteria do not. We talked about how that results in a high mutation rate in the DNA, bacteria and archaea. Eukaryotes have a nucleus that protects their DNA, which helps minimize mutations of DNA. So how did the nucleus originate? Where did the nucleus come from? We think, this is our leading hypothesis, long time ago, in a galaxy far, far away, no, just kidding, in a galaxy here on planet Earth, the ancestor of today's eukaryotic organisms, plasma membrane, just kind of infoldings of the plasma membrane started to surround and enclose the chromosomes, the DNA. Alright, so we don't really know how this happened. This is our best guess again. So if this infolding of that membrane, and that's the membrane surrounding that DNA, was heritable, meaning that that characteristic could get passed on in the DNA of that organism, and it was advantageous, then what do you think natural selection would do? Natural selection would probably select for that characteristic, right? And part of the advantage of having a nucleus is, one, protection of the DNA, right? Now we have an extra layer surrounding the DNA, so it's protected from environmental factors that could cause mutations, right? But it also allows for processing of genetic information. So we haven't covered this yet, but prokaryotes do not splice their RNA. They do not, so whatever is on their gene in that piece of DNA gets transcribed and translated into protein. In eukaryotes, we regulate that, what parts of a gene are transcribed and translated. And we can do that because the processes of transcription and translation are separated by this nucleus. Okay, so this allows us to alter what parts of a gene end up in that RNA and then end up in that being used to build a protein, right? So based on how we splice, based on how we rearrange parts of that RNA that's made in transcription, we can get lots of different proteins from one gene. Okay, it's called alternative splicing. So separating out the process of transcription and translation means that we can make more diversity of proteins, which is pretty handy. If your main goal in life is living and reproducing, right, having a wide variety of proteins that you can make by separating out these processes is definitely going to be advantageous. Okay, let's talk about the mitochondria. Where do the mitochondria come from, right? So most eukaryotic cells have hundreds, if not thousands, of mitochondria in each cell, pumping out ATP. That's their entire job. They're just ATP factories. But mitochondria is weird because it has those two membranes, right? It has a huge internal surface area. So where did the mitochondria come from? So again, mitochondria make ATP, that energy currency of the cell. They're surrounded by two membranes. Big deal. They contain a circular chromosome. Does that sound kind of familiar? And they make their own ribosomes. And those ribosomes in mitochondria are different than the ribosomes that we find in the cytoplasm of the cell. They're totally different. The ribosomes inside mitochondria are more similar to ribosomes of prokaryotic bacteria. Also, the other crazy thing is that mitochondria are not assembled. They just go through binary fission. They basically split and reproduce on their own. So our cells do not assemble new mitochondria. We just inherit the mitochondria from our old cells as those mitochondria split. Okay, so how do mitochondria originate in eukaryotes? So Lynn Margulis is the person that is credited with really pushing this endosymbiosis theory. It was around, this idea had been around for a while, but it kind of been considered like a fringe idea. Like, no, there's no way that that's actually how mitochondria got into eukaryotic cells. But Lynn Margulis, one, carried out some additional experiments, and two, put all of the evidence together and put all the reasoning together and said, no, come on, this makes sense, that this is how this would have happened. Okay, so basically the idea is that long time ago, an archaean cell would have engulfed a bacterium cell, We think something related to what we know of as today is an alpha proteobacterium, but for some reason, I'm not sure why, that bacterium was not digested. It's remained to live inside of that archaean cell, okay? So it just came to live inside, endosymbiosis, inside living is basically what that means. So that bacterium just started to hang out and live with that archaean cell inside of its cytoplasm. And then the descendants of that bacterium are mitochondria that we have today. Okay? So that bacteria has been dividing for billions of years, and it's residing in our cells today. We think that this maybe happened about 1.8 billion years ago, roughly. We don't know, right? Because again, this kind of stuff doesn't fossilize well, but that's our best guess. Okay, so a lot going on here. Mitochondria, some of the main lines of evidence for endosymbiosis theory of mitochondria. It's surrounded by two membranes. Bacteria have two membranes, right? They contain a circular chromosome, like bacteria, okay? They make their own ribosomes. They used to be independent cells, and those ribosomes are very similar to the ribosomes that we find in bacteria. They're different than the ribosomes that we find in other eukaryotes. They split by binary fission to produce new mitochondria. They're not assembled like the Golgi is assembled or the ER is assembled. No, mitochondria reproduce on their own. And alpha proteobacteria are still around, right? And we think that that would have been a sister group or sister taxa to the bacteria that ended up living inside of this archaean cell. The archaean cell that engulfed that bacteria and then went on to kind of fuel the rest of domain eukarya. So here's a eukaryotic tree, all right? Most of these eukaryotes have functioning mitochondria. Some of these guys up here have lost their functional, they have mitochondria, but they're not functional anymore. So we think that that was a trait that would have been inherited from the common ancestor of all of domain eukarya. Again, these two branches, mitochondrial function is lost, but they have some remnants of mitochondria. So that tells us that that trait of having a mitochondria living inside of your cell would have been inherited by the common ancestor to all of domain eukarya. Okay, now what about chloroplasts? They're kind of similar to mitochondria. I mean, mitochondria make ATP by breaking down sugars, right? But in organisms that photosynthesize, they use chloroplasts, the organelle chloroplast, to capture sunlight and turn that solar energy into chemical energy. So this is in land plants, red algae, green algae. Their chloroplasts have a double membrane. Does that sound familiar? They have circular chromosomes that are separate from the chromosomes that are in the nucleus. And the chloroplasts, just like the mitochondria, go through that process of division to produce new chloroplasts inside the eukaryotic cell. They are not assembled. Chlorophyll A is the primary pigment molecule to collect sunlight, to absorb that solar energy. And they use water as an electron donor in photosynthesis. So we haven't talked about photosynthesis yet, but basically, in the process of photosynthesis, water is broken apart to steal the electrons off of it. So that's what we mean here. And these chloroplasts release oxygen as a byproduct of photosynthesis. Okay, so it's a very specific chemical process, biochemical process that's going on inside of chloroplasts. And we see it somewhere else in the world too. Again, our friend Professor Lynn Margulis, says, hey, it makes sense. We could apply this idea of endosymbiosis theory to chloroplasts as well. Single ancestral celled eukaryote that already had mitochondria. Okay, so we already had a eukaryotic cell with mitochondria inside. That proteobacterium that was living inside, now it's just a mitochondria. We think that it probably engulfed the cyanobacterium. Cyanobacterium was not digested. Sound familiar? Cyanobacterium went to live inside of the eukaryote, happily just hanging out inside that cytoplasm. And then the descendants of that cyanobacterium are now what we have, chloroplasts. Okay, so cyanobacteria are gram-negative, got that double wall going, right? Remember the cell wall of gram-negative bacteria? have two cell membranes separated by a layer of peptidic glycan. Okay, so here's, again, cyanobacteria. We've seen similar species like this. Okay, so very similar lines of evidence for the chloroplasts. Chloroplasts have a double membrane, just like cyanobacteria do. Chloroplasts have a circular chromosome, just like bacteria do. They split to produce new chloroplasts. They're not assembled. Okay, they go through binary fission. Chlorophyll A is the primary pigment, use water as an electron donor, and release free oxygen, just like the process of photosynthesis in cyanobacteria. It's possible, but very, very, very unlikely for this chemical process to basically occur identically in two separate cells. If these were not related, if cyanobacteria were not the sister group to chloroplasts, that would be highly unlikely. Okay, it's possible that they could have settled on the same idea, but it's more likely that cyanobacteria are the sister taxa to the chloroplasts that ended up inside of eukaryotic cells. So chloroplasts are a little different. We have two primary and secondary endosymbiosis processes. So we think this lineage right here, Archaeoplastida, engulfed a cyanobacterium, and we call that primary initial symbiosis because the chloroplasts in these organisms have only two membranes. Okay? But there are some chloroplasts that have four membranes. Okay, so this is where friend Sarah P. Gibbs comes in. She was investigating some additional types of chloroplasts and found, hey, that's weird. These chloroplasts in these lineages have four membranes, but these ones in these other lineages have only two. What's going on there? So brown algae and diatoms in the Chromalveolata, they have four membranes to their chloroplasts. Four membranes covering their chloroplasts, not two. Okay, so how did that come about? Well, this is what we call secondary endosymbiosis. So we think that a predatory eukaryote, again, just a eukaryote with mitochondria, would have engulfed a red or green algae that already had a chloroplast inside of it. So this is another eukaryotic organism right here. It engulfed that eukaryotic algae, but it was not digested. And then the nucleus in the algae was degraded. And then now this chloroplast has 1, 2, 3, 4 membranes, that fourth membrane being the vesicle basically that was used to bring it into the cell. Okay, so that's our guess as to how those four membraned mitochondria came about, is this process of what we call secondary endosymbiosis. So we think that happened. The euglenozoans have chloroplasts that have four membranes. Okay, and they're also mixotrophic, meaning that they eat and photosynthesize. They consume other organisms and they photosynthesize. We also think that with the red algae over here, diatoms, brown algae, and then the dinoflagellates, epicomplexans, that they would have eaten a red algae that was already a eukaryote and that was not digested. So these guys over here also have 4 layers to their chloroplasts. So we think that there were 2 independent secondary endosymbiosis events. But regardless, all photosynthetic eukaryotes share these basic components. They all use primary pigment of chlorophyll A. Water is the electron donor for photosynthesis. PSS is photosynthesis. And they release free oxygen, just like cyanobacterium. These are the key events engulfing a mitochondria, engulfing a proteobacterium, engulfing a cyanobacteria, and then secondary symbiosis engulfing a eukaryotic algae, and we get 4 membranes, right? So that's where the membranous organelles come from. The nucleus, again, remember, comes from the infolding of that cell membrane surrounding the DNA, would have been heritable and advantageous.